Hi everyone, welcome to my chemistry channel. In this lecture, we're going to discuss about the nitrogenous enzyme, which is used in the nitrogen fixation in plants. And nitrogen is a very essential element, which is very full element for build up the amino acid protein molecules in living system. In many of the systems, that is, if you see the structure of proteins, amino acids, and certain porphyrins, which is a macrocyclic ligand, all these are incorporated by the nitrogen element. In earth, the natural element, the nitrogen exists as a dinitrogen, as a gas. So in this nitrogen is utilized in the all forms. So first this nitrogen is converted into a specific form which is useful for the all living system. Okay. And basically this nitrogen is very active, very inert also. This nitrogen which is exist in the earth is very inert. So it chemically very inert because of ionization potential, first, and having high bond dissociation energy. The bond dissociation energy nearly in 945 kilojoule per mole, and low electron negativity, and the electrons from the outer shell of the nitrogen cannot be removed easily. Either by adding the electrons or removing of electrons is not very easy in the case of nitrogen. So electrons very inert. So due to this reason, this nitrogens cannot be converted into easy form. So it exists as a natural dinitrogen gas. So it can be converted into another form by using the two main process. So this nitrogen is converted into ammonia by using the two main process. First one is a biological process which is facilitated by the bacteria, and the second in artificial manner or industrial manner. Through the well-known process called Haber-Bosch process. Haber-Bosch process. Okay. So in plants, the ammonia is very essential compound to synthesize the amino acids and proteins. So plants need to synthesize amino acids and proteins with the help of ammonia. So this ammonia is prepared through nitrogen molecule with the help of some bacteria present in the plant. So that is called biological process. In industrial process also this ammonia is made. Okay, let's see that. First see, in industrial process, in artificial manner, the ammonia with hydrogen at 400 to 500 degrees Celsius at 200 atmosphere with the help of either iron or molybdenum catalyst, which produces ammonia. So this is called Haber-Bosch process. During this reaction, the energy will be 50 kilojoule per mole. So during the reaction, if you see the mechanism, it has two intermediates. If you draw the energy diagram with reaction coordinate delta H, it forms the two intermediates This This is the nitrogen reactant and this is product. During that it has two intermediates. This two intermediates is the one is diazine, another one is nitrogen. So these are the two intermediates are formed during the synthesis of ammonia industrially, and these two intermediates have high energy compared to reactant and products, which has less energy compared to these things. So this reaction is kinetically favored, but not thermodynamically. Okay. So it was synthesized the ammonia by using Haber-Bosch process. This ammonia is used to synthesize the fertilizers, which is for the plant growth and decrease the soil for infertility. Okay. So this ammonia is used to produce the fertilizers. So this reaction is kinetically favored but not thermodynamically favored. So this is about the industrial process of ammonia production. So when we see for the biological synthesis of ammonia, in biological system, certain bacteria contain blue-green algae. Blue-green algae. So this algae present in certain bacteria. In biological system, there are two types of nitrogen fixation is happen. One is symbiotic nitrogen fixation, and it's a non-symbiotic nitrogen fixation. What do you mean by symbiotic and non-symbiotic nitrogen fixation? Symbiotic means the bacteria is present in the plant itself. That is, the bacteria present in the host plant. So this nitrogen fixation is happened by the bacteria present in the host plant. Okay. A non-symbiotic nitrogen fixation is made by bacteria that are present in the soil. They are independent. Okay. So these non-symbiotic bacteria are independent and these bacteria are dependent to the plant itself. You see the example for this. The well-known bacteria Rhizobium, and for non-symbiotic Clostridium pasturinium, Acetobacter, Granulobacter. And these are the example for non-symbiotic bacteria which is not dependent on the plant cells, so which is living in the soil, which is used to convert the atmospheric nitrogen to ammonia. Okay. And if you see in symbiotic bacteria, example Rhizobium bacterium, which is found in the root nodules of leguminous plants. Root nodules of leguminous plants such as beans, cloves, peas and soya. So these are the plants which are having the Rhizobium bacteria in the root nodules. Okay. So this bacteria fix the nitrogen to ammonia in the root nodules itself. Okay. So this is the symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. So this mainly the symbiotic nitrogen fixation is very essential and it is happened by the Rhizobium bacterium. Okay. So this rhizobium bacterium has certain enzyme called nitrogenase. Nitrogenase enzyme. So this nitrogenase enzyme converts the atmospheric nitrogen into ammonia. So this nitrogen, in the presence of nitrogenase enzyme, which act as a biocatalyst, which facilitate the reaction and converts the nitrogen to ammonia. Okay. And if you see the nitrogenase enzyme, this is in the 3 types. Okay. First one is the molybdenum nitrogenase, and second one is the vanadium nitrogenase, and third one is the iron nitrogenase. Okay. Basically, this nitrogenase enzyme have 2 types of metals, 2 types of metalloproteins present in the nitrogen enzyme. That is, so one metal which act as a cofactor, which is iron, act as a cofactor in all this nitrogenase enzyme. So in here, molybdenum iron nitrogenase and vanadium iron nitrogenase and iron nitrogenase. Okay. Among this vanadium nitrogenase is a very less activity, having less activity compared to other nitrogenase enzyme. In this structure, you see it has 4 Fe and 4 sulfur units. 4 Fe and 4 sulfur units. Among these 3, the molybdenum nitrogenase is a more efficient nitrogenase enzyme which facilitates this reaction, which have high activity. Okay. High activity. Let's see the molybdenum iron nitrogenase. Structure. If you see this structure, it has 2 types of protein, having 2 types of cluster. One is the iron small protein, two forms. Another is the molybdenum iron protein. Okay. And if you see the molecular mass, this is a small protein which having the molecular mass of 60,000 units. Okay. It's composed of 2 Fe4SO4 units. Okay. Let me see the structure of this. And if you see the molybdenum iron protein, the molecular mass is around 220,000 to 240,000 units. So this is the molecular mass of molybdenum iron protein. And let's see the structure of molybdenum iron protein. First, the small protein iron protein. Okay. The 2 Fe4SO4 units here. So the 2 Fe4 units are connected by cysteine groups, 2 cysteine groups, and this 2 sulfur is connected by the sulfur bridge, and it has cysteine unit. Okay, this is the iron protein which having the two Fe4S4 units. These two Fe4SO4 units connected by the two cysteine groups and above and below, and these two sulfurs are connected, and one of the unit has two cysteine units. Iron atom has two cysteine units and one serine units. In another unit only it has two cysteine units. So this is about the structure of iron protein. And if you see them, molybdenum iron protein structure, it's also having the similar to the structure which having the two Fe4SO4 units. Cysteine units. This iron are connected with the cysteine groups. Okay, so the two Fe4SO4 units are connected by the molybdenum Here the nitrogen is bind on the molybdenum. Okay. So if you see the molecular mass of molybdenum iron protein, it's around 2 lakh to 20,000 to 2 lakh to 40,000. So it is made up of two Fe4SO4 units, and one of the iron atom from one unit and another iron atom from another unit combined with the molybdenum and has two sulfide bridges. Okay. Here the iron atoms are connected with the cysteine units in the one Fe3SO4 units. Okay, and here this is the active site. So this nitrogen is bonded with the molybdenum. So the molybdenum is the active site of this nitrogenase enzyme. Okay. So the reaction is facilitated by using some electron transfer agents. So first, the reaction is proceeds like molybdenum. This is the active site. Transfers the electrons. So finally it synthesizes ammonia and leave this from the molybdenum, okay, and stored in the plant, which is for the amino acid and protein preparation. So for this, the source of electrons is from the electron transfer agents. So first, the electron transfer agents, the source of electrons from the oxidation of pyruvate. In plants, the oxidation of pyruvate takes place, which transfers the electrons to smaller proteins, pyridoxine and flavoprotein. So these are some electron transfer agents in the plants which facilitate the electron transfer chain reactions. It will transfer the electrons to the smaller protein, that is P cluster. So this is called P cluster iron protein. This iron protein transfers electron to the molybdenum iron protein and facilitates the chain reaction which leads to the production of ammonia. Okay. So the nitrogen, the plants, the simple schematic representation of ammonia production will be okay. So the energy for this reaction is given by the ATP. So the ATP is converted into ADP. So the energy from this reaction is given to this ammonia production. Okay, so this is the overall reaction of this nitrogenase enzyme. Okay, and this nitrogenase enzyme, the ammonia production from this nitrogen, this is a symbiotic nitrogen fixation. In symbiotic nitrogen fixation, the production ammonia which is used to convert to amino acid, proteins and urates, which is from the symbiotic nitrogen fixation. Okay, and non-symbiotic nitrogen fixation also gives the ammonia, which is converted into nitrites and nitrates. Okay. And these nitrates and nitrates are formed by using some nitrifying bacteria. So first the ammonia from the non-symbiotic nitrogen fixation is converted into nitrite using some nitrifying bacteria such as Nitrosomonas or Nitrosococcus. And this nitrite further converted into nitrate using nitrifying bacteria called Nitrobacter. Okay. So this is the nitrogen fixation in symbiotic and non-symbiotic system. So symbiotic nitrogen fixation, the production ammonia is converted into amino acid, proteins and urates, and from the non-symbiotic nitrogen fixation, the produced ammonia is converted into nitrates and nitrates using some nitrifying bacteria. So this is the overall reaction of this nitrogen fixation. So this nitrogen fixation needs energy. So this energy is given by the reaction of hydrolysis of ATP to ADP. So here the 11pi is the inorganic phosphorus. Okay. So this is about the nitrogen fixation in plants. I hope you all understand. Thanks for watching. If you like my video, like, comment and share my video. For more videos, please click the bell icon.